I'm Jeremy. I'm a professor at UCSF. I work on cilia and how cilia are useful as signaling organelles. Who wants to go next? Savannah, you're next to me on my screen. You want to
Hi,
kick us off?
I'm Savannah. I'm from UC Davis. I'm a fourth year. I work in Dr. Sheila David's laboratory, and we study DNA repair and enzymes that facilitate that repair.
Awesome. You're gonna have to ping someone.
Oh yeah, the classic Zoom circle. I'll go with Lena.
Hi, I'm Lena. I'm from UC Santa Cruz. I'm working on how antibodies target the virus spike.
Awesome.
And I picked Megan. Is that how you say your name?
It's A. But, so my name is A. I am a master's student at San Francisco State University, and I'm currently doing my research at UCSF at the Institute of Neurodegenerative Diseases. And my research focuses on understanding protein aggregation, tau phosphorylation, and how it affects aggregation of proteins in neurodegenerative diseases.
Awesome. Who are you working with, eh?
Dr. Daniel Southwood.
Okay, fantastic. You're gonna have to pick on somebody, eh?
Let's go with Kathy.
Hi, everybody. I'm Katie. I'm a first-year PhD student in the neuroscience program at UCSF. And so since I'm a first-year student, I'm still doing my rotations between different labs. And so I'm broadly interested also in neurodegenerative diseases and cellular and molecular mechanisms behind those.
Awesome. This has got to be a weird year to be a first-year student, Katie.
For sure, in a lot of ways, but we're banding together and making it work.
Okay, good. I'm glad to hear it. Are you willing to let the cat out of the bag? Are you leaning in any particular way towards one rotation over the other?
I am, but I'm not gonna reveal quite yet. But
okay,
I'm very strongly leaning towards one lab, but I'm starting my last rotation now, so I'm trying not to jump the gun quite yet.
I hear you. Yeah, no, it's good to keep an open mind. I agree.
Yeah, exactly.
Okay.
Yeah.
Well, awesome to meet you, Katie.
Yeah. I can popcorn it next to Max.
Hey everyone, my name is Max. I'm a fourth-year grad student in the neuroscience program at Davis, and I work in the Olson lab, where we research psychedelics and their related analogs and how they can be used to treat neuropsychiatric conditions. My projects particularly focus on the molecular mechanisms through which they can promote neuroplasticity.
Can I ask an indelicate question, Max?
Yeah, go for it.
In a psychedelic lab, in a lab that studies psychedelics, do the participants in the lab also participate in the ingestion of said psychedelics?
You mean like us, like the grad students? No.
Okay.
No.
Clinical trials.
It's a super like, I mean, everything's under a lock and safe and it has to be logged in and out. It's very, yeah, no, you can't, you can't sneak a little for yourself.
Gotcha. Yeah. Cool. You wanna ping either Robert or Hirsch?
Let's go with Hirsch.
Hey everyone, I'm Hirsch. I'm a second-year biophysics grad student at UCSF, and I work on like engineering immune cells, novel functionality into immune cells to make new therapeutics.
Awesome.
I'll jump in at the end, as I also work in the same lab as Max, and as the DEA person in charge of keeping the regulation all on the books and up to snuff. There, it is highly regulated, and yeah, of course, no tampering from the lab mates.
Yeah. I mean, yeah, it looks like they're going to do a recording, and so we are being recorded at this, which is an interesting segue into a discussion about psychedelics and personal versus research uses of psychedelics. Yeah, I guess not to belabor this point, but I do think that there was probably a looser era in the generation prior to mine in the experimentation on psychedelics. Robert, Max, does the name Alexander Shulgin mean anything to you guys?
The godfather of psychedelics.
Yeah, yeah. I met that guy a couple of times. He was up in Marin, I think. And yeah, I guess he's no longer with us.
How did you meet him? Was it at like a conference or something?
It wasn't actually. It was just hanging out here in San Francisco as a graduate student. That guy was around. So like, I don't know, he would go to like, I don't know, concerts and things like that in San Francisco. And if you were kind of hanging around, you would kind of run into this old guy. Yeah, he went by Sasha. Yeah. But
Incredible.
Yeah.
Yeah, very cool.
Yeah. Just for a little background for everyone else, Alexander Shulgin, he's like super famous for synthesizing like hundreds of novel psychedelics, and he would test them on himself and his friends. And he like rediscovered MDMA and some other notable psychedelics.
Definitely some good science there. I mean, he's written a few books, and I've taken some chemicals from his books and made analogs of them as a chemical biologist in the lab. Max is more on the neuroscience lab, neuroscience part of the lab, and I'm more of like a chemical biologist making.
Oh, fantastic. Are you guys in the same lab?
Yeah.
Okay.
So
Okay.
he does a lot of like the rodent behavior and some of the cell culture work, whereas I try to make photo switchable tools. So it's like compounds that photoisomerize, and we can have an active form and an inactive form to really try to understand and probe mechanism of action, as well as various brain regions, how they're facilitating some of these therapeutic benefits, as well as like hallucinogenic profiles.
Awesome. Yeah. 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 Sasha gave me a copy of Pikal once. So yeah.
We definitely have a copy in the lab.
Okay. Awesome. Okay, so we don't only have to talk about recreational drug use. I guess we could talk about other things too. Yeah, I don't know if we're going to get a cohort of people to mingle with, unfortunately. And alas, I'm going to have to leave in about 10 minutes to go host a celebration for Gita Narlikar, who people at UCSF might know, who just got inducted into the National Academy of Sciences this morning. So, so an important thing. I don't know. Somebody have a pressing question, or Lena, you want to tell us about astrovirus? I've never even heard of astrovirus before.
Yeah, it mostly affects children. It causes gastroenteritis, and we're studying how antibodies target the spike, and we found that antibodies target several different regions on the spike. So it's unknown the cell receptor binds, where the spike binds the host cell receptor. So we're thinking that antibodies block the receptor binding site, or maybe there's sites that either one can be blocked.
Cool. How about you, A? You want to tell us about tau?
Sure. So I mean, in neurodegenerative diseases, as some of you may know, protein aggregation is a major area of research that people are studying, and tau is one of the proteins that is prone to aggregate in the neurons, specifically in the intracellular part of neurons, and what people have found is hyperphosphorylation is a major factor that is found in these aggregated tau proteins. So right now I am trying to figure out and to have a better idea of whether phosphorylation is definitely is really driving this aggregation at all. I mean, I believe there are other cellular factors that are involved in it too, but phosphorylation could be one of the factors that is driving this aggregation. So we are basically using phosphorylated protein expressed from eukaryotic insect cell systems and using that to see how the aggregated tau proteins, how the structures looks like compared to unphosphorylated tau, which is expressed from E. coli cells. And so far, the when we use phosphorylated version of tau and use it in protein aggregation studies, and the structures that we are getting out of it is really similar to the patient protein aggregates or filaments specifically from Alzheimer's disease. So yeah, so right now there's some challenges that we are having right now is in order to move forward with looking at the structure of these protein filaments, tau protein filaments by cryo-EM, we need a large volume of the filaments, concentration of filaments, and that's with the current method that we are pursuing, we're not getting a lot of filaments. So right now we are trying to optimize the current method to see how to push forward that to do cryo-EM and study the 3D structure of these protein aggregates, tau protein aggregates.
Awesome.
Yeah.
Thanks, eh? Yeah, tau seems super mysterious, like an awesome hallmark of Alzheimer's, but whether it's pathogenic or not wasn't totally clear to me. Is that still not really clear?
Yeah, it's still a big question that still needs to be answered. But right now what we are really doing is just at the basic level, we are not even understanding how this whole protein aggregation is happening. So I think that's the first step to, kind of, uncover the story of how this whole thing aggregation is happening in the brain. Yeah.
Awesome. Well, do you guys have anything you want to talk about, or should we ask Savannah to tell us what G quadruplexes are?
I could totally talk about this for a little bit. But I also was going to say, I do want to hear, so there was like a list of questions or whatever. I would like to know how everyone's research was affected by COVID, like at each of the schools. So we can talk about that after I tell you about G quadruplexes. But my research focuses on a family of enzymes known as NEAL, and these enzymes can remove oxidative damage from the genome. And they're really unique in that they can remove damage from these stacked structures that form in DNA that are extremely unique, basically all guanine residues that stack in layers. And they're known to be of interest in gene expression. So there's a lot of ideas that they can be targeted therapeutically. So if a G4 folds or unfolds, it can turn on or off a gene. So you could use that as a modulator for gene expression. And so what's really interesting about my enzyme is that it can remove damage from these G4s and hence saving their structure in a way. So our research looks into the details surrounding this enzyme removing damage from these structures. So yeah.
Awesome. Yeah, I didn't realize we had some scripted questions that we might go through, but yes, I would love to hear how people's year has been affected. I mean, Katie already touched upon the fact that it's a very strange year to be a first year. Yeah, I got to imagine it's got to be a very strange year to be a grad student, period.
Yeah, I feel like at Davis. Sorry, Katie. I feel like
you're
at
good.
Davis it was like we were locked down for like till, I don't know, maybe RJ and Max can speak on this, but I think it was like 3 months. And so we were kind of worried that we would never get to go back to lab ever. You know, it's like, oh my gosh, how long are we going to be out of here? But I was actually kind of surprised that we got to go back in like May or June, but we only got like 2 people at a time in the lab, and we have like a 14-person lab. So it's like you're going in like maybe once every other week or something. It was crazy at first. I don't know if that was the experience for other labs.
It was a good time to catch up on reading and writing. So fortunately, you know, I was wrapping up a few projects, so it really blocked out some time for me to write, and that was good. As well as transitioning into new projects, you want to make sure you're doing all the proper your due diligence ahead of time to understand where the field is and before diving in. And that was it actually, you know, silver linings, it worked out all right for my project in its current stage.
Awesome. I'm glad to hear it, Robin. How about you, A? How was it?
Yeah, right now, at least at our lab, it seems we're pretty much back to normal. The only thing that's still really affected is trying to order things like gloves. Those take a long time to come in.
Yeah.
Yeah, there's been like a lot of shortages on a lot of stuff lately. It's ridiculous.
Pipette tips are so hard to find. It's like a scavenger hunt every time.
I hear ya. Yeah, we use these Transwell filters for some of our specialized tissue culture, and they went from like back ordered 1 month to back ordered 2 months to back ordered 6 months to back ordered a year. I don't know what. Yeah, they must have been on the evergreen or something like that.
Yeah, it's the same thing in my lab. Like from the regular tips, now it's kind of transitioning to pipette, like filter tips, which is a little more expensive. And like right now we are like just buying whatever tips we can get from and all the brands. Like, so right now in the lab, we have like sorted brands that we don't even know which one is gonna fit with our pipette tips sometimes. It get confusing.
Yeah, I have heard, but I haven't tried this yet, that you can take if you've got the Rainin pipetters, and getting those tips is what's I think especially difficult right now, especially the P1000s. Like you can take the barrel off of the Rainins and 3D print a different barrel that then you can use other manufacturers' tips on.
Wow.
That's dedication.
Yeah.
That's ambitious.
Yeah.
Yeah,
Well,
you have to have a 3D printer though.
yeah, if it's that or mouth pipetting, then you probably, yeah.
We're going back to the old school way.
Exactly. Yeah.
I think it's important to notice
That's so funny because we have a ton of Rainin tips, but we only have one Rainin pipette.
Yeah.
I think we have to pass around to use the tips.
Oh, no.
And we don't have any other brand tips that we actually.
Awesome. Awesome. Okay, well, sounds like things are going to go for another 8 minutes. I unfortunately am going to leave you talk amongst yourselves. It was wonderful to get to hear a little bit about your science. Be well, everyone.
Thanks, Jeremy.
Thank you.
Thanks.
Thank you. I think it's also super important to realize, you know, and with how scarce resources are and how things can be costly now, and time is so valuable, how much, you know, we must appreciate the ARC's support and funding during this time frame, right? So that's always useful. Hirsch, great.
Yeah, I agree.
Awesome.
Thank you. Thank you.
Yes, thank you very much, ARC's funders.
RJ, are you our question person now?
Yeah, so we kind of talked about the different protocols for COVID at various universities. It seems like, you know, everyone's work kind of got put on hold a little bit, but there are definitely some silver linings. What about some really big questions in your field? So I know everyone has those pie in the sky questions that you'd love to answer. Where is the impact of your research, and how do you guys see making a difference in the field? And if no one wants to start, I will happily jump in.
I think in like the field of like neurodegenerative diseases in general, which is, you know, kind of my interest, is that what we really want to know is like, I think a really big question is what are like the precipitating events for a lot of different diseases. So like right now there's a lot of current research on like disease progression or like trying to, yeah, mostly in progression, trying to like stop the disease from progressing. But in a lot of cases, we don't know like what the initial insult is. So for example, I just finished a rotation in the lab that's studying Parkinson's disease. And we know that Parkinson's disease is largely due to like death of like a specific class of neurons in the brain, but we don't know why those neurons are dying. We're trying to understand why they're vulnerable, but we don't know, you know, what is the precipitating event that started that. And I think that would be, I mean, that would be like groundbreaking if people could find that out, not just for Parkinson's, but for, you know, any given disease, and not even necessarily just neurodegenerative diseases, but yeah.
I have a quick question about that. So like, in looking for these events, is it mainly like environmental stuff, or is it genetics, or both? Or like, is there like a, I don't know, leaning more towards one or the other, or is it kind of still up in the air?
It's, I mean, you probably also know a lot about this, but it's interesting that for a lot of neurodegenerative diseases, something like 90% of them are sporadic. So we don't necessarily know, you know, you can't nail down what the genetic component of it is. But, you know, that means for a remaining 5 or 10% that there is, you know, like a known genetic contribution. So I think that kind of suggests that there's, you know, likely both are at play, that maybe you have a genetic mutation that makes you especially vulnerable, but, you know, there are likely environmental factors that are at play, or, you know, lifestyle differences between people that could make you susceptible.
I completely agree. So like for tau specifically, there are definitely some disease mutations that are found in different neurodegenerative diseases, and they are prone to be like, they tend to have aggressive aggregation compared to a regular like wild type form of tau protein. And yeah, just like Katie said, like definitely there's some genetic factors playing in to accelerate the disease progression, but I feel like there's a large chunk of it is also like environmental.
It's really interesting to me that there's, you know, I feel like for most classes of diseases, the pressure, like especially the selective pressure that drives the disease pathogenesis is really obvious. Like in cancer, in infectious disease and viruses, it's really obvious that there's like a clear pressure for these to develop. But degeneration, as far as I know, like it's not that obvious what the, maybe it's a thermodynamic thing. Maybe there's like a propensity for protein systems as they get more complex to find these, you know, minima that are categorized by these aggregates. But it seems really complex, and we don't even understand the, you know, fundamental basis in a good way.
Yeah, along those lines, even just like chronic stress. So our lab, the Olson lab at UC Davis, focuses a lot on depression, and just chronic stress can, you know, make these neurons in the prefrontal cortex atrophy away from each other. And now that they're, you know, atrophying, there's less, it's like a positive feedback loop in a bad direction, where it makes it harder and harder to overcome these barriers and regrow these brain cells for these neuroplasticity events. And that's really where we're trying to spearhead our science through these psychoplastogens, and actually trying to make like non-hallucinogenic analogs of these compounds that can regrow brain cells. So understanding how these systems work and how these compounds are facilitating this regrowth of these essential neurons in the brain, that's where we'd love to see advancement in the field. Truly understanding the mechanism of action. All right, let's do a nice question about future self. So where do you all see yourselves in 5 to 10 years? Are we all an academia bunch? Are we some industry people? Where do we see each other, or see ourselves, I guess?
5 to 10 years is kind of a long time, but you know, one would like to hope that I'll have a career. So I think that I'm not really an academia person. I think I want to try to go more like the industry route. To where yet, I do not know. But I think the cool thing about having the ARCS fellowship has not only allowed for exploring alternative, you know, scientific, I guess, routes, but also, you know, being at a university system, they provide you with opportunities to explore other careers as well. So I will have to see where that leads me, but I definitely think that I do want to move into a more industry area.
I'm just curious, who here does want to stay in academia, or is maybe like leaning towards it post grad school?
Looks like we're an industry bunch.
No one?
you
Damn.
say who?
Okay.
I think you. I
Yeah,
think you
sorry.
said that you were leaning. Yeah.
Oh, no. Sorry. I just wanted to get it like here amongst us.
You want to call out the?
I was just curious.
No, I think I definitely would want to be leading some kind of research group. Like, I think like that academia is like the classical setting to like lead a bunch of people like with some research objectives. I think it exists in industry and other places as well, but I think that's what I value the most, and that's what brings me leaning, into academia more and more.
I'm so thrilled by like the speed at which, industrial settings can pump out research and really advance the field. So I'm excited to see the other side of the coin. And hopefully in 5 or 10 years, I'll be in an industry setting.
Yeah. But I think it's also.
I'm hoping to do industry too.
Say that again. Oh yeah.
I like the applied nature of the work.
Yeah. I find it very interesting to see where like I think there's some disciplines where academic labs like give industry a run for their money, and I always find that incredibly shocking. That the budget scale is an order of magnitude different at times, but academic labs can still compete. But obviously that's I think the exception rather than the rule in many fields.
All right, team. Well, we got a chat message saying we are free to end anytime now. So it was awesome getting to meet you all and hear about your research. Hope everyone has a great day, and thank you, ARCS.
Yeah, it's good to meet everyone.
Nice
to meet you guys.
Nice meeting everyone.
Good job on everyone's poster.
Have fun.